Here we will start with, as I was telling you, that we will start with very basics. Like protein structures are very important because they interact and they communicate, and there is chance that there are targets. Protein interactions to how to do molecular visualization, and molecular visualization is very important for cheminformatics or for drug discovery. So all of you who would be joining the program we'll be learning about different molecular visualization tools and how to utilize them. Then we will go into cheminformatics. First, we learn about the macromolecules, and then we learn about the small molecules and how to explore the chemical space, like databases, prepare for virtual screening, large databases, how to utilize them for drug target identification or for searching similar ligands. Those are the key things. And then key businesses. So there is always a business component or industry component associated with our training here. So then the focus is to go into those databases, explore those databases, learn about accessing those databases, and curate bioactive molecules and find the right kind of molecules. So that could be specific, for example, kinase inhibitors, or you're looking at very specific type of inhibitors with specific type of biochemical or physiochemical properties. So in this, we will be, again, the hands-on part is there, where we will be learning about the softwares, tools, which are freely available for you to utilize and use them along with the mentor or after the mentor session, and practice on your own, because we will be also providing the recorded session and the content and the material and the links in a systematic manner, so that you learn from the basic. And even if you miss some of the sessions, you can go back and you'll have it all with you to learn and do it anytime you wish to. But it's advisable to go along with the sessions, so that the desired outcome can be you can get the desired outcome within that time space. Now, exploring the protein data bank. The protein data bank I think is the cradle for structural biology and for drug discovery, structure-based drug discovery. So here we find all the experimental structures, and you can see that as many as 60,000 structures are available for humans, and the list goes on. So we have a lot of structures now compared to the previously, and you can see the number of structures that are deposited and how they have grown over the time. And different, like for example, SARS-CoV-2, just in a few years, like 3 years, there are more than almost 3,000 structures, which is massive in understanding about a virus as small as SARS-CoV-2 and having so much structural information. So just imagine if you can understand the system biology from that, how many opportunities are there to help. So yeah, I mean, this is the repository where we will be finding the structures and understanding the very basics of structure, how to understand the structure, how to change something in there, how to incorporate mutations, how to understand the role of the mutations, and exploring different types of research projects and structures. So here, the participants who will be joining, they'll have the opportunity to explore the three-dimensional space and find their own structure of interest, disease of interest, and work with that structure to further investigate and find out how they are folding, how they are maybe functioning, what are the key residues, where to find the active site, what are the solvent accessibility areas, if there is an allosteric site or a conventional drug discovery will work, what are the different types of drug discovery approaches one could take, whether it is a flex, whether it is what kind of docking are we going to talk about, what kind of simulation. So all those things are possible. So we will elaborate on each of these sessions. For example, target structure prediction. I have already discussed a lot about it, so we'll not discuss it further. But these will be discussed in the context of a case study. So where we will be covering these different topics, and then we will be working in that demonstration section in the during the session. So different types of modeling, what are the different technologies that are used, what are the different tools that are used, what are the pros and cons of using one before another, and what are the most popular and validated methods that are out there. Then target identification and virtual screening. So virtual screening is again very important in that drug discovery aspect because it's being used. So Prashanjit has a question: which software do I need to install on my Mac? So for molecular visualization, Prashanjit, you can download Chimera or Pymol. Chimera is free and a very powerful tool. Pymol is proprietary, but also for academic, there are free versions which are available. So I think you can start from there and have a molecular visualization tool for sure installed in your software, in your MacBook to start with. So now about virtual screening, as I was telling you that virtual screening, so let's simplify this. So virtual screening aims to computationally search the universe of potentially organic molecules to identify a manageable number of virtual hits, right? So what it is doing is that it is searching for molecules which could be considered as hits. So identifying them in the context of how the initial screening could be done. So one such computational method which we use for virtual screening is docking, right? Molecular docking. So it uses the three-dimensional structure of the target protein and places small molecules into the binding site. So there is, however, a computational complexity which is there in docking. The evaluation of each small molecules require consideration of its low energy three-dimensional conformation. So this is what I was telling you about before, that when we are doing virtual screening, when we are starting from virtual screening, there are different steps. So one is virtual screening, another is then intensive docking, which is where it is computing all the different parameters which are also important, which is not done at the screening stage, because screening, you have a large library. So generating low conformational three-dimensional calculating the energy of that conformation is not possible at that point of time. So we have tools which do that and help us screen from larger library to smaller libraries. But obviously, what is the best approach in bioinformatics for me is to utilize multiple tools and then come up with a table which has the top scorers from each of those tools. So that in a way, within bioinformatics, validates our work. So that will be covered in the 5th session, and then we'll learn about the different steps, and we have some projects that we have already worked on. For example, this is a drug discovery project that company has worked on with Roswell Park Cancer Institute, which we will be using as a case study. And then we have the actual session on the other aspect after virtual screening, the in-depth docking and stages of drug discovery process. So we will learn about the pharmacological aspect and drug discovery aspect and the different conformation using the top softwares which are freely available, but also the concepts are like the logic is same. The software might vary, but once you know the logic, once you know how to utilize it, what are the different aspects, I think the language remains the same. So it's important to understand the basics and also the parameters that you have to look into when you're looking into these different aspects. So one of such experiments, if you do with us, you will be able to explore on your own, and also we'll be able to help you in case you use different softwares of your own interest. So we till the 6th session, as you could see, we are learning about the basics, and we are learning from a very beginner aspect to for those who are learning for the first time or who want to learn from the very beginning and basic level to the advanced level. I think first to 6 sessions are to help them reach at that stage, and then we go into more advanced topics such as data science and AI in healthcare, cheminformatics using R, these such topics. So in this aspect, I would like to talk a little bit about QSAR. So QSAR or QSPR, these are based on the idea that the structure of a molecule might influence its physical, chemical, and biological behavior. So we have been using, by the way, QSAR for last 40 years in the practice of agrochemistry, pharmaceutical chemistry, toxicology, and many other facets of chemistry. So QSR has effectively modeled many physiochemical properties such as toxicity, metabolism, interaction between drugs, carcinogenesis. So initial QSR model such as Hansch and Free Wilson analysis, very popular, I guess, uses that use regression models to relate potency and structural and chemical properties such as chelation, substitution patterns, surface activity, hydrophobicity, solubility, electronic forces, electrostatic forces. So in the context of the overall protocol for constructing QSR model for drug designing has been systematized, and many open source tools and softwares are available for such steps which consist of several modular steps for chemoinformatics and machine learning techniques. So in QSR modeling, you start with molecular encoding, in which the chemical structure of the drug molecule is used to derive the chemical features and the property, and then you go on and work on that. So there is an opportunity for those who want to learn about this in the advanced modules that we have prepared, followed by the case study. So we have a case study which is the discovery of sirotinib kinase inhibitor for lung cancer, and also there's a blockbuster drug that recently got there's a company that was recently got acquired by BMS. BMS is a Bristol Myers. It's a very big pharma company for $4.1 billion. So and it's a inhibitor. It's a kinase inhibitor, and it works on different chemotherapy treatment. So I think, I mean, that's some of those case studies are really helpful for understanding that what are the success stories, and also what are the different ways these companies have utilized to reach at that stage. So, I mean, tools and softwares, here's a list from Autodoc to molecular docking to ab initio modeling. So we will be able to utilize many of these for the understanding, unless there are proprietary, which is like Desmond, Schrodinger, or Charm. So other than that, the ones that are available, we will be able to use for the project work. If you have a hypothesis, if you have a structure that you have started working with, or if you have a disease type that you are working on. So those who would be joining, so we have different modes of joining the program. So one could choose to join between for 1 month or 3 months. So for 3 months, we will be able to work on research proposals, and then we also have the research fellowship program, which actually helps students to complete a research project. So then, as I was telling you, that this entire drug discovery is focused on the modern approach. So you can work on oncology related projects, you can work on infectious diseases related projects, and you can use multiple types of data types to be able to come to the drug discovery or drug target, or you can use several other approaches that we can work with. So it's up to you, and it's also you will be also learning about these approaches. If you have not if you think that you're new to all of this and you're not ready for it, so that's why the beginner level is there, so that you are ready for all of this. So again, here the focus is AI in molecular drug discovery. We discussed about artificial intelligence playing a role, and also deep learning has a huge role. So we'll discuss all those aspects in this session. And then understanding that from the point of view of how these different technologies. So Google has DeepMind program, has the open AI program where the Dr. Puneet just discussed about chat GPT. So incorporating the new technologies, the real like really impactful technologies that are came into 2023, 2022. So I don't think there are any other programs which utilizes that, but this is what we've been doing. We are updating it like every month, every week, and so that we can enable those who are joining to get to the latest technologies, learn about the latest advancements, and apply them into their research. So this will include like understanding, and this would include pharma and bioinformatics industry integration. This will include like different types of softwares for molecular visualization. This will include advanced cheminformatics, which is to further manipulate the structures. This will include the research projects that I've been discussing about, and this will include integration of omics data in research projects, if that is what your direction is. It will include you to work on pharma projects, which is for specific pharma problems. It will include for you to work on different infectious disease projects that are there, for example, SARS-CoV-2, and learning about different binding properties. We have already published articles on malaria vaccine design and resistance using popular population genetics and drug discovery and molecular dynamics on all of that. So in the end, we have a case study which is on successful as well as the failed drug development, to give you an insight about how what worked and what has not worked. This is a recent publication that we were able to complete through this training, and it's an infectious disease genomics research which was recently published in Molecules. So I think that concludes my presentation for today. And we have still the registration and the cost and the scholarships, and really exciting time, because we have a really great scholarship model which is just available for next for few days, which is I think what Sparsh will be talking about.